This may sound silly, but the chapter 6, the chapter on infection, is one of my favorite chapters in the book. I just think microorganisms are really interesting. If you've had a chance to take microbiology, then you'll know what I mean. Microbiology is one of the most fun biology classes you can take because you get to grow stuff. It's cool, and you get to classify it and figure out how to stain it and how to kill it. So it's a really interesting field. But basically, microbiology is the study of microorganisms or microbes, which are very small living forms. And they're visible only under a microscope. So microorganisms include bacteria, fungi, protozoa, and viruses. And many of them can grow in an artificial culture medium. So that's what you get to do in microbiology lab, which is super fun. So we have different classifications of microorganisms, non-pathogenic, which are ones that don't usually cause disease, and under certain conditions they might. We have a lot of microorganisms in our body which are considered part of the normal flora of our body. So they live with us, and they do a lot of things for us too, really. Really, if it weren't for microorganisms, it would be very difficult for us to sustain life as we know it. Non-pathogenic microorganisms are often beneficial. Pathogens, on the other hand, are disease-causing microbes. So sometimes they're referred to as germs. Infectious diseases result from an invasion of the body by microbes and multiplication of the microbes followed by damage to the body. So the microorganisms, they cause spoiling of food and other things, but they also cause, a lot of the foods we eat, like some cheeses are developed by microorganisms, like Swiss cheese and blue cheeses are developed by microorganisms. Wine and beer, yeast is a microorganism. So there are a lot of things that microorganisms do for us. And so if you have those same microorganisms, they're great when you put them in cheese or in wine or beer, but if they got in the wrong place in your body, they could be pathogens. And we often we call those pathogens, those microorganisms that are beneficial in the right place, but they're baddies when they get in the wrong place, we call them opportunistic pathogens, meaning given the opportunity, they could easily cause you disease, but most of the time they're doing their own thing and they're helping us out. So kind of interesting. So here's the little graphic from the book with all the cute little types of microorganisms, and we will talk about them. So on the top we have a bunch of different bacteria. On the bottom left we have a virus. On the bottom right we have a fungus or a yeast. I guess it's a chlamydospore. So it's a fungus basically. So bacteria are one type of cell, and then the yeast and fungus are another type of cell. And viruses actually aren't cells. They don't have a cellular mechanism. So we'll talk about all the different types. So bacteria are single-celled organisms. They're classified as prokaryotes, meaning they don't have a nucleus. They have no nuclear membrane separating their DNA from the rest of the cell. So as human beings, we're eukaryotes. We have a nuclear membrane that separates our DNA from the rest of the cell. So bacteria just hang out and they have their little metabolic processes, and they reproduce by binary fission, where they split in two. And they have a very complex cell wall structure, and that's their protection from the outside world. And they're individually living. They don't require our living tissues to survive. So they can live in ponds and food and in the environment. They don't require living tissues to survive, and they vary in size and shape. So this is our little binary fission. So the DNA replicates, the cell divides, and you have two identical daughter cells. And then each daughter cell divides and you have 4 identical daughter cells. So that's how bacteria reproduce. The major groups of them are classified by shape. So bacilli are rod-shaped, so they're the little green ones in the middle there. Spirochetes are spiral forms. Cocci are spherical forms. So you get individual ones. You get diplococci, which are two little spheres that hang out together. Streptococci, which are long strings of spheres, and staphylococci, which kind of clump. So they're basically the groups. They're grouped by shape and kind of who they hang out with. So the basic structure of a bacteria, regardless of its shape, is it has a rigid cell wall that protects and provides that specific shape. So the cell wall is what determines the shape. There are 2 chemical types, and we call them gram-positive and gram-negative. And it depends, that classification goes by whether or not you can stain them with a certain stain, and that is determined by the chemical composition of their cell wall. So it's useful in selecting appropriate antimicrobial therapy because gram-positive organisms are susceptible to antibiotics that don't kill gram-negative organisms, and vice versa. The cell membrane is located inside of a cell wall, and it's selectively permeable. Just like our cells have selectively permeable membranes, bacterial cells also have selectively permeable membranes. So we have a nucleus holding our DNA, but bacteria's DNA is just kind of hanging out in the middle there, and there are a bunch of ribosomes floating around in the cytoplasm, turning the DNA into proteins. We also have plasmids, which are little individual loops of DNA, and those can be acquired from other bacteria, and a lot of times plasmids convey antibiotic resistance. So that's where we get into trouble with antibiotic resistant bacteria. So a lot of times the effect of an antimicrobial drug or an antibiotic has to do with the cell wall. So the antibiotic has an effect on the bacteria's cell wall. So in the book they talk about a lot of different groupings of bacterial cells, and we're not going to worry about that or the individual parts of the bacteria. We just kind of generally want to know about them. So one of the reasons why, besides the development of antibiotic resistance, one of the reasons why we can use certain drugs to kill bacteria is because they are prokaryotes. There are drugs that can be used for fungal infections and other things that attack eukaryotic cells, but that's a problem because it also could damage our own cells. But different antimicrobial drugs have different effects. They can have an effect on the bacterial cell wall. They can interfere with the bacteria's metabolism. They can interfere with the bacteria's DNA and protein synthesis, or they can cause damage to the cell membrane. So those are kind of the 4 main mechanisms of antimicrobial drugs. So the bacteria has what we call the slime layer, which I think is a great name. It's an external capsule, and it's not found in all bacteria, but it's found in some of them. It's outside the cell wall, and it offers them additional protection. It's an additional barrier to the outside world. Sometimes they have flagellae, which are little projections that are attached to the cell wall, and they provide some motility. So it's like a little tail that they can use to propel themselves with. Pili or fimbriae are little hair-like structures found in some bacteria, and they assist in attachment to tissue. They are also can be used to transfer DNA to another bacterium, so that's how those antibiotic resistance plasmids get transferred. So the cell membrane is inside the cell wall. It's selectively permeable like our cell membranes, and the cytoplasm contains one long strand of DNA. So instead of having a bunch of chromosomes like we do, they have one long strand of it. It also contains ribosomes and RNA, so it can translate that DNA into proteins. And then it has those plasmids, which are the DNA fragments not attached to the chromosome, and they are exchanged during conjugation with other bacteria. And often that's where the antibiotic resistance comes from. So, gram-positive bacteria produce exotoxins, which are toxins that they release into the environment. Endotoxins are present in the cell wall of gram-negative bacteria, and they're released on the death of the bacterium. So those are the things that can cause septic shock, and it increases our capillary permeability, and it can cause infection to spread. Bacteria also have enzymes that can damage tissues and promote the spread of infection. So when they're the pathogens, they have some bad little tools that can mess us up. Some bacteria can form spores, and it's species specific. And the spore is basically a dormant or latent form of the bacterium, and they can survive long periods in a spore state without needing as many nutrients. They're highly resistant to heat and to disinfectants. So you can have this sort of latent bacterium lurking, resistant to heat and disinfectant, and then later on it comes out to get you. So viruses, they are small, obligate intracellular parasites, meaning they need a cell in order to reproduce. They're parasites. They can't reproduce on their own. They consist of a protein coat, also known as the capsid, and the protein coat comes in various shapes or sizes. Viruses can mutate quickly. That's one of the reasons why you can catch a cold and give it to somebody else and it's different. So for their nucleic acid, bacteria always have DNA. Viruses can have DNA or RNA. And their classification depends on which nucleic acid is present in the virus. So if they have RNA, they need something called reverse transcriptase in order to convert the RNA to DNA and use our cellular mechanisms to reproduce themselves. So that's one of the ways that when they're trying to come up with drugs that combat retroviruses, which are the RNA viruses, they a lot of times will use things that target reverse transcriptase to keep the virus from reproducing. Cool, right? So here, like all these different viruses, fancy ones, and I think I have some pictures in here that are micrographs of viruses. So really, the virus is basically that protein capsid is a shell for their RNA or DNA or for their genetic material, and a way for them to get it into another cell and use that cellular mechanism of the host cell to reproduce itself. So in an active viral infection, so aren't these just the creepiest looking viruses? But the virus attaches to the host cell, it injects its genetic material into the cell, and the viral DNA or RNA takes control of the cell, so it's like a takeover. A hostile takeover. It uses the host cell to synthesize its own proteins and nucleic acids, and then the new viruses are assembled by the cell, and then it bursts out of the cell, breaks it open and kills it basically, and releases those new viruses. So viral infections, that's where your symptoms come from that lysis of the cells. So here's our little virus. It comes along, it attaches to the host cell, it uncoats itself and it injects its DNA into the nucleus. It uses the host cell to synthesize its components, it assembles new viruses, and then it shoots them out of the cell. So viruses can, in a latent viral infection, the virus enters the cell with an active infection, and viral proteins are produced and inserted into the membrane of the host cell. So this can stimulate an immune response and destruction of the host cell. But if you have a depressed immune system for whatever reason, the virus can actually reproduce actively, and the immune system doesn't react to the host cell. So it can just sit there making more viruses, which is not good.